In our previous video, we had a discussion on the disease that is candidiasis. So today we will discuss another fungal disease that is rhinosporidiosis. So before having a detailed discussion on the disease, let us have a look on how this word rhinosporidia has been evolved. Basically, this rhinosporidiosis is a disease which is caused by the agent or the pathogen that is Rhinosporidium siberi. The word Rhinosporidium includes two words, that is rhinos and sporidium. Another word is siberi. Siberi is an honor for the scientist who described this organism in 1910, and the scientist was Guillermo Siber. So to honor him, the word has been added to the pathogen's name as Siberi. Now, the rhinos is nothing but a Greek word rhinos, which means to say that it is a nose. So basically, in this disease, the affected human beings will have a characterized nasal polyps in their nose. So therefore, it is called as the rhinos. And when it comes to the sporidium, the nasal polyps, which are having a structures inside those tissues, so they look like a spore-like structures under the microscope. Therefore, it is called as sporidium. So when we combine these three words, now the organism is called as Rhinosporidium siberi, which literally means a spore-producing organism if discovered by a Siber in the nose of the human being. So this is about how the name of the disease has evolved. Now let us discuss about the history and the habitat of the rhinosporidiosis. When it comes to history, we already discussed it. In 1910, a scientist named Sieber discovered this Rhinosporidium siberi in human being, and later it is also recognized in animals such as cattle, horse, mules, and dogs as well. Initially, it is thought that it is to be a fungus, but later studies have showed that it is an aquatic protestant parasite. So, but as far as veterinary related exams are with respect to veterinary field, in this context, it is grouped under the mycosis only. Now, let us discuss about the habitat of the Rhinosporidium siberi. Usually, these organisms are found in the stagnant water, and this disease is very endemic for the countries like India, Sri Lanka, Argentina, and some African regions. And the infection is acquired through mainly the activities such as bathing or drinking from the contaminated stagnant water. And also sometimes the traumas or aberrations for the mucous membranes will act as a portal of entry of the organisms. So these are the key points with respect to the history and habitat. This organism has been discovered by the Sieber in 1910. This organism has been discovered in the human case, and the source of infection is usually it is stagnant water, and the main hosts are in cattle, horse, mules, dogs, and also humans. Now, let us discuss about the pathogenesis of the disease. So, exact mode of transmission is not well studied or not understood. In this disease also, it has been noticed that there is no any contagious spread of the disease, which means to say that there is no proof of animals to human or man to man or man to animal spread has been noticed. And a key important point in this pathogenesis is the injury sites or wound sites act as a portal of entry for the organisms through these traumatized tissues, and environmental source such as the organisms can be carried by the soil or dust particles. So inhalation of those contaminated soils, dust or air might be the possible route of the infection. So this is about the pathogenesis. So here you can observe this is the respiratory epithelium, and inside the tissue here you can observe the structure. This is the sporangium, which is filled with the different sized sporangiospores. So these spores act as a source of infection. So this is a whole sporangium which is filled with the sporangiospores. Here nose showing this is the nasal polyp, and the small dots are the sporangiospores. So here also you can observe this is the sporangium which contain these minute small spores. These are sporangiospores. So these act as a source of infection. So here also this is the sporangium containing sporangiospores. Now we'll discuss about the pathogenicity of Rhinosporidium siberi. In pathogenicity, there are two important things, and those are the mode of entry and transmission of the organism. The habitat of the organism is usually in soil. Therefore, water acts as an essential tool for the transmission of the organisms, and through the wounds or in aberrations which were on the skin, that is nothing but the traumatic inoculation will act as an portal of entry of organisms into the body or tissue of the affected host. Now, the infection stays localized to the mucosal epithelium. Therefore, this infection is usually not any systemic. Now, let us discuss about the life cycle of the organisms inside the host tissues. So, usually these organisms will develop as the endospore. Endospore is round in structure, which ranges from 6 to 10 micrometer. So this endospore will grow and become into a thick-walled sporangium. So this will be sporangium. So this is of size 100 to 400 nanometer, which contain thousands of endospores inside it. So these are the endospores which are inside the thick-walled sporangium. So this is the immature sporangium. Now, this immature sporangium will become a mature, and in the mature sporangium, a characterized pore is created. Through this pore, the mature endospores, these are nothing but the mature sporangiospores, are released. And again, these released sporangiospores will act as a source of infection for the other tissues. Again, they start replicated in the different tissues. So therefore, that leads to the tissue hyperplasia, and also immune responses are noticed in the host affected. So this is what mature sporangiospores will escape via pore in the sporangial wall. Now, let us have a look on the clinical features and the lesions of the disease. These common lesions are commonly noticed at the sites such as the nose and nasopharynx, and also very important site is lacrimal gland. The lacrimal gland are also a very important site of the infection or the lesion. Now, how these lesions will appear? So they have gotten characteristics features, and they are look like a polyp structure. Therefore, they are called as polypoid growth, and they are usually pink to red in color, and they look like a strawberry-like structures. They are sessile and are pedunculated in structure. They are highly vascular and feeble. So therefore they are feeble, they bleed easily whenever they are touched. So this is about the clinical features and lesions. Now let us discuss about the different symptoms of the disease. The disease is characteristically it is chronic in nature. So it takes much longer time for their growth in the tissues. It has to pass through multiple life cycle of the organisms. Then they invade and again they start growing in the tissues. Therefore, it takes much longer time for the production of the disease. It is called as a chronic infection. And usually these polyps are painless in nature. Nasal bleeding is very common. And as these polyps are there in the respiratory tract, so therefore there is a which leads to the obstruction for the normal respiration. So in such condition, there is a high chances for the chances of asphyxia is noticed. And also these polyps may also extend to the oral cavity. So this is about the rhinosporidiosis in other species. Now, when it comes to the rhinosporidiosis in dog, the nature of the disease is zoonotic potential. So dogs can be a source of the transmission to human beings, and this is very rare disease, and it is also a chronic fungal-like infection which is caused by the Rhinosporidium severi. The common sites in the dogs are also the nose and nostrils. Sometimes the eyes are also involved. The structures such as the nasolacrimal lesions and the conjunctiva of the eyes are involved. The clinical signs such as respiratory signs, as we discussed earlier, sneezing, wheezing, labored breathing, that is difficult in breathing, and nasal bleeding, that is epistaxis, are also involved. And the growth and lesions looks like the cauliflower-like masses in the nostrils are visible, and their polyps or growths which looks like white or yellowish in color, and they are spotted appearance, that is due to the fungal endospore. These polyps growth which were there in the tissues have a fungal endospores. Therefore, they look like a spotted appearance. So this is the spotted appearance of the nasal polyps or any other different tissue polyps. So the key features are the lesions are polypoid, they are vascular and feeble. Therefore, they bleed easily, and they resembles as a strawberry-like polyps. Now, this is the last part of the video. Here we will discuss about the diagnosis of Rhinosporidia. Usually, there is no culture is available because Rhinosporidium severi cannot be grown in artificial culture. Now, therefore, the different definitive diagnosis is only possible through the demonstration of spores and sporangia. So, this demonstration of spores and sporangia can be done via the wet mount technique. So, in this technique, the nasal discharge is collected, and these discharges are smeared on the slides, and if stains are prepared, then stains can also be applied, and cover slip is put on the slide, then directly visualized in the microscope. So, this is a direct wet mount technique, and sometimes the histopathology of the polyps can also be done through the tissues collection and stained with the H&D stains. Now, in the wet mount technique, the microscopic features are, when it comes to spores, they are in the size of 6 to 7 micrometer, and they grow gradually. There is enlargement of the tissues, enlargement of the spores are noticed, and these spores are again developed into sporangia. These sporangia are around 100 to 300 micrometer in diameter, and they have got, these are the spores, and these spores will become as the sporangia. They have got this thick wall, and this wall is made up of cellulose and chitin, and these sporangia contain different spores, and they may range up to 16 to 20,000 spores are there in each sporangia. So again, there are different stages in the tissue sections are noticed. The tissue sections may be of the young tropic form, or mature sporangia form, or collapsed sporangia may be visualized in the microscope. So when it comes to the young tropic form, it is usually the immature sporangia. Immature sporangia, which is of small in size, and these the endospores are also not matured. So it is a very growing form of the sporangia. This young tropic form will again convert into mature sporangia. These are larger size when compared to the previous entropic form, and they packed with the multiple spores are there. And again, this mature sporangia will open as a pore, and via this pore, these endospores, these are nothing but the mature sporangia spores will release or escape. So in such condition, this sporangia will collapse. So such a section can also be visualized in the microscope, and the characteristics pore in the sporangium wall can also be visualized. So this is about the microscopic feature of the sporangia. Now let us come about discuss about the treatment of rhinosporidiosis. Usually, the surgical intervention is suitable, and surgical excision of the polyps is the only possible technique for the treatment. And the drawback of this technique is that the high reoccurrence is noticed, and till now there is no effective antifungal or antibiotics are available yet. So this is about the disease rhinosporidiosis. It is a very small disease, so be thorough about this. Now we'll discuss some of the MCQs based on this topic itself, so that to make it more concrete to understand. Now let us discuss a few MCQs on this topic, that is rhinosporidiosis. What is the most common site of infection for rhinosporidiosis? Rumen, hooves, nose and pharynx, nose and nasopharynx, liver. Just now we discussed, the answer is nose and nasopharynx. The characteristic lesion of rhinosporidiosis is best described as a dry, scaly crust on skin, a strawberry-like febrile bleeding polyp, a deep, non-healing ulcer, multiple pustules over the body. The answer is a strawberry-like febrile bleeding polyp. Now, the causative agent of rhinosporidiosis, Rhinosporidium severi, is best classified as an bacterium, fungus, parasitic protist, and virus. The answer is that is parasitic protist. So this is the most accurate answer. One can also go with the fungus, but most accurate is the parasitic protist. How is rhinosporidiosis most commonly transmitted? Through inhalation of spores, through the ingestion of contaminated feed, through traumatic inoculation during the contact with the stagnant water, through the direct contact with an infected animal. The most appropriate answer is C, that is traumatic inoculation during the contact with stagnant water. So here one can get confused with respect to the through the inhalation of spores and ingestion of the contaminated feed. But the thing is that when it comes to most appropriate, we had already discussed that the stagnant water act as a medium for the transmission of the disease, that is transmission of the causative agent. So therefore, through traumatic inoculation during the contact with the stagnant water is the most appropriate answer for this question. Now, the major clinical sign associated with nasal rhinosporidiosis is profuse diarrhea, persistent coughing, epistaxis, lameness. The answer is epistaxis, that is nasal bleeding. So these are few MCQs on this rhinosporidiosis. So in next video we'll discuss another fungal disease. So from this side only, but thank you.